Good evening, everyone. This is Dr. Jess Armine, coming to you from the Office of Methylation Support at the Center for Bioindividualized Medicine here in southeastern Pennsylvania. Tonight we're going to talk about reading your 23andMe without losing your mind. At the Center for Bioindividualized Medicine, we find answers through genetics and integrative medicine. And in attendance here is Dr. Jess Armine, yours truly, and Shawn Bean. Introduction. The purposes of this presentation is to give the reader a general idea of what the polymorphisms, otherwise known as single nucleotide polymorphisms or SNPs, mean and how you may determine if they are expressing in you. Please know that the presence of SNPs do not mean that there's a problem in the indicated pathway. The absence of SNPs do not mean that the pathway is working normally. The SNPs may or may not be expressing in you. This is why it is advisable to consult a healthcare provider who understands the SNPs and to help put it all together. This is an informational lecture. The information may or may not pertain to your condition. Treatment should be properly done on an individual basis in consultation with your healthcare provider. There are numerous interpretations of the SNPs. This informational lecture is from my own personal research, knowledge, and experience. There may be other differing interpretations. Permissions. This study will look at one of my patients utilizing the raw data from 23andme.com and Sterling's app version 1 at mthfsupport.com. Understanding that upon this date, Sterling has a version 2 app, but I thought it would be good to go over the basic information prior to tackling that one. The patient has given me his kind permission to share his findings with you. All of his identifying information has been deleted. The research for the SNPs was done utilizing snippedia.com and genecards.org unless otherwise indicated. And by the way, if you happen to be treating the SNPs incorrectly and you look to the right, this is what could happen. So, let's go. So, what are these genes and SNPs that we are looking at? Well, a gene encodes an enzyme. Enzymes run the metabolic processes in the body. The presence of a polymorphism might indicate that the enzyme encoded by that gene may not be working at its usual efficiency. A normal gene, usually you'll see negative negative or the color green, means that the enzyme is working at its usual efficiency. Heterozygous, plus minus, which is usually written as yellow, indicates that the enzyme may be working at about 60% efficiency. Homozygous, plus plus, which is also seen as red, indicates that the enzyme is working at 10 to 20% efficiency. Think of it this way. Think of the biochemical pathways as highways that are able to process a certain level of traffic to produce their stated result, whether that's detoxifying, creating glutathione, manufacturing excitatory neurotransmitters, manufacturing ATP, or whatever it happens to be. The normal expression, the green, is like an 8-lane highway. The yellow is like a 4-lane highway. And the homozygous or red is like a 2-lane highway. If you don't have a lot of traffic, it doesn't matter whether you're green, yellow, or red. It's not going to slow down. Everything's going to be fine. But if you try and push 12 lanes of traffic through an 8 or 4-lane highway, the processes will slow down, and you may or may not get the results you're looking for. Pushing 12 lanes of traffic through a 2-lane highway, let's face it, that highway is going to slow down or crash or not give you what you're looking for. What is this traffic I'm talking about? The increased traffic comes from chronic infections, viral loads, food allergies, leaky gut syndrome, immune upregulation, autoimmune diseases, fungal or yeast, parasites, or any combination of those. So, everybody's always asking me about the SNPs and how to look at them. So I'm going to give you the principles in interpreting the polymorphisms or SNPs. Interpreting each SNP one by one is courting confusion at best and mental illness at worst. Each SNP is but a part of a biochemical pathway designed to create a certain result. It's best to view the SNPs from the 30,000-foot point of view. I get that from the corporate types. Okay, they like that phraseology. Therefore, we're going to look at the SNPs in groups, not necessarily individually. One last thing. When you're looking at your 23andMe, and if you've done Sterling's app, you'll see various columns. Okay, the first column on the left is the gene and variation. The gene is that CYP1A1. In this particular case, we're talking about the cytochrome P450 pathway, which is part of phase 1 liver detox. After the little star, the gene has a particular variation, okay, which just makes it slightly different. The RSID number is, if you were going to research it on your own, is how you would find it. You would click this or copy and paste it, and it would take you to SNPpedia. SNPpedia is the Wikipedia SNPs, and trust me, it takes a while to figure out what the heck is going on. The risk allele. It determines whether your gene is a polymorphism or not. It took Sterling and her research group like between an hour and 10 hours per gene to figure out what combination of amino acids would portend a polymorphism. My suggestion is that you simply accept the fact, okay, and look at the gene and look at the results. If it's green, there's no polymorphism. If it's yellow or red, there's a polymorphism. What that means, we're about to see. First page. This is again is the cytochrome P450 pathway, which is your detoxification pathway in your liver. If you were to mentally bend this page in half, the polymorphisms above that imaginary curve have to do with estrogen dominance. This is the kind of person who may have a lot of PMS, PMDD, may have estrogen or hormonal difficulties. In this particular case, the CYP2D6 is homozygous, and this individual may have difficulty breaking down things like dextromethorphan, Robitussin DM, beta blockers, antiarrhythmics, and certain antidepressants. The CYP2D6, the next one, the T2850C, sometimes this can result in an ultra-fast metabolizer phenotype. This is the kind of person that metabolizes their medicines or vitamins very, very quickly. This is the kind of person who has side effects from all their medicines, not because they react to the substances, but because they metabolize them so quickly that they get a very, very high level of medication, but they don't get the therapeutic duration. To digress for a moment, when you take a medication or a vitamin, you're supposed to get a certain blood level for a certain period of time. That's called the therapeutic duration, the therapeutic level, and the therapeutic duration, and then it's supposed to metabolize out. In the ultra-fast metabolizer phenotype, they get it all at once, get the side effects because of the high amount, and then shoom, it goes down, and they never get the therapeutic duration, hence it never helps them. This kind of person, if this is expressing, should be taking medicines or vitamins frequently, okay, in smaller doses than one big dose. On the next page, you see what I like to call the Gs. GPX, GSTM, GSTP. This is all the glutathione pathway. So if you have glutathione and you have polymorphisms in this pathway, it may predict how available the glutathione is to your body. Glutathione peroxidase may change oxidized glutathione back to reduced glutathione. Glutathione S transferase influences asthma risk. There's lots of different things that you'll see if you look these up, but the general takeaway here is this is how your glutathione pathway is going to act. The NATs, which means N-acetyltransferase, the first thing you think of is how it may break down aldehydes. What are aldehydes? Think of formaldehyde. Okay, we all know what formaldehyde is, and it smells terrible. We all know that if we drink too much alcohol, we're going to get a hangover. Well, ethanol becomes ethylaldehyde, which becomes ethylcritic acid and carbon dioxide and water. And even though that's under lactic dehydrogenase, okay, these are the enzymes that it works through. So if they're not working, you're going to get stuck in the aldehyde stage. And ethylaldehyde is only one carbon off of formaldehyde. And let's face it, you're not going to feel good drinking alcohol. If you develop a yeast infection, okay, especially a large intestinal yeast infection, yeast will produce a ton of acetaldehyde. Again, very much like formaldehyde. And let me tell you a little secret. In the 1980s, when they were going, don't feed the yeast, don't feed the yeast, don't feed the yeast, okay, they were absolutely correct. Because with a lot of yeast, there was a lot of acetaldehyde, and a lot of acetaldehyde could cause all kinds of havoc in the body, including things like MS or headaches or migraines and just everything. So that is the indicator here that this person may not be able to break down their aldehydes. SOD do. One more time, Jess. SOT2, superoxide dismutase. Superoxide dismutase is what breaks down free radicals. Okay, and it's important for mitochondrial dysfunction. So when I start seeing them grouped like that, I begin to wonder if this person may have mitochondrial dysfunction. PON1 is specific for organophosphates, pesticides, things like Roundup or glyphosate, which I know I'm pronouncing incorrectly. Okay, and we did a podcast, which will be available soon, on just that subject. Okay, little clinical pearl for everybody here. If you wanted to give yourself superoxide dismutase, you could grow organic wheatgrass, and it's not hard to get. And you get from Amazon, I guess, you can get a wheatgrass juicer for about $20. And as long as when you grow the wheatgrass, it doesn't grow above 14 inches, okay, you're not getting any gluten in that. So you can grind that stuff up, give yourself about 2 ounces of superoxide dismutase away. I guess I meant to say a day. Yes, a day. You can't buy this in the store because the enzyme breaks down very quickly. Tongue tie, cleft palate. Dr. Ben Lynch has done a great amount of research in this area and has developed some treatment ideas. If you go to the indicated website and then click Educational Tools, you will find a download concerning what you can do if you have an indicator of tongue tie or cleft palate. This is especially important if there has been tongue tie or cleft palate in the family already. So I encourage you to download Dr. Ben's treatise on the area because he's got some great ideas. Allergy and mold. The next couple of pages have to do with allergies and mold, and they're not exactly well constructed, so I'm going to work you through them. These first 2 HLAs have to do with mold. They're not super exact, but the fact that there's a polymorphism, you put it in the back of your brain that this person may not be very friendly with mold. You see IgE on the bottom. It really belongs on the next page. If you look at the set of SNPs there, these are the IgE SNPs, and they are very significant. And IgE is that immunoglobulin that will end up in releasing histamine. And oh my God, this person's going to produce a ton of histamine. Why is that important? Well, when you have a lot of histamine, you have a lot of ripping open of cells and leaking of cells. That's why when you have a histaminic reaction and you want to blow your nose, you get all those noises that sound like what I just made. Okay, think about what's happening all over your body. Every time this person gets an antigen and it goes down this pathway, he's going to produce a prodigious amount of IgE and a prodigious amount of histamine. The IgG, which is usually from food allergies, okay, also looks significant. And on the next page, you have IgA, again, more food allergies. And what is the takeaway here? With this type of pattern, if this patient develops leaky gut syndrome, he or she is going to produce a prodigious amount of antibodies. The more antibodies you produce, the more inflammation you're going to have. Okay, and that's the bottom line. That's why us alternative medicine practitioners tend to go after leaky gut all the time, because it's the greatest creator of inflammation. And that's where the rubber meets the road. That's where the body starts deteriorating. Clotting factors. We can go into a bunch about the clotting factors, but here's the takeaway. If you're a bleeder, you'd already know about it. The problem here is, are you clotting too quickly? If you nick yourself shaving and it takes you 10 seconds to clot, that's not a good thing. If you're a woman who is cycling and you're passing a lot of clots, okay, that's not a good thing. Why? Because when you're clotting too quickly, you can set yourself up for heart attack, stroke, and the like. Methylation. The first four there I like to call the A's because they have to do with angiotensin and hypertension. And you can see that the angiotensin converting enzyme, the adductin, the acetyl coenzyme A acetyltransferase, and the AGT on the bottom all have to do basically with control of the lumen of your angios, which are your veins and vessels, and that controls your blood pressure. I copied this picture from Neurological Research Institute, which is Dr. Yasko's group, and I tried to expand it and add a few things. This is a rather simplified version of the folate methylation, methionine, transsulfuration pathway, and the biopterin pathway. And I'm going to talk you through it. Okay, let's start in the middle circle at THF. And by the way, if I say something fast, it's meant to be ignored. The THF represents your dietary folates, and that's called tetrahydrofolate. It passes by an enzyme called MTHFS, which is methylene tetrahydrofolate synthase, which then creates 5,10-methylene tetrahydrofolate. The 5,10-methylene tetrahydrofolate passes by the dreaded MTHFR, otherwise known as the mother-father gene, or worse, which really means methylene tetrahydrofolate reductase, which then takes the 5,10-methylene tetrahydrofolate and turns it into 5-methyl tetrahydrofolate. We head north to MTR and MTR, where it becomes methionine and methyl B12. For reasons that are not clearly understood, the methyl B12 tends to lose its methyl group and has to be remethylated by SAMe, or through that BHMT pathway where trimethylglycine becomes dimethylglycine. In that part of the pathway, you can see where methylation contributes to RNA and DNA and protein synthesis. Mind you, I'm being very, very, very simplified. It's a whole lot more complicated than this. SAH is formed, and then AHCY, which creates homocysteine. And then we go down past cystathione beta synthase, which creates cystathione, and passes by another enzyme to create cysteine, and with alpha-ketoglutarate becomes glutathione. From CBS on down, of course, you have SUOX that takes sulfite and turns it into sulfate. From CBS on down, it's called the transsulfuration pathway, and its purpose is to create glutathione. If you have blockages or problems here, you may have a higher ammonia and a lot of brain fog. If we go to the left side, where you see DHPR there, which is the biopterin pathway, which MTHFR does in fact have some contribution. This is where tyrosine becomes dopamine and tryptophan becomes serotonin. These are broken down by monoamine oxidase and COMT, which is catechol methyltransferase. And we see a lot of polymorphisms in these guys, which is rather important because it tells you how much excitation someone's going to have. Okay, you also see on the bottom the GAD SNP where glutamate becomes GABA. The body's interesting. It's intricately simple and simply intricate. So I leave this with you to look at as a referent. As we're looking at this page, you're seeing that HCY causes the hydrolysis to adenosine, to homocysteine, and is thought to play a critical role in the regulation of biologic methylations, according to Dr. Yasko. The BHMT changes trimethylglycine to dimethylglycine, providing a methyl group. Sometimes this person will have inner sadness. Dr. Yasko feels that this BHMT is the essential shortcut through the methylation cycle, and again is helping to convert homocysteine to methionine. The activity of this gene product can be affected by stress, by cortisol levels, by many things, and can play a role in ADD, ADHD by affecting the norepinephrine levels. As I noted before, CBS is not a radio station, but is cystathione beta synthase, and is important in the transsulfation pathway that produces glutathione. One of the things you've got to ask people is, do sulfur foods bother you? Garlic, onions. Okay, and I'm not talking about just producing a little gas, I'm talking about making you sick. And here's a clinical pearl for everybody out there. Somebody with a lot of CBS genes should be careful, at least be cognizant, of juicing. Juicing's good for you, right? Juicing's excellent. But if you're juicing a bunch of sulfur vegetables, you're going to give yourself a lot of sulfur. Think about this. Think about this analogy. You go to a vendor, it's summertime, you want yourself a nice glass of fresh squeezed orange juice. So you ask him for the orange juice, he's going to take 6 oranges, slice them open, squeeze them, hand you the orange juice. Well, I submit that you probably would never sit down and eat 6 oranges, but there you are drinking everything but the rind of 6 oranges when you're drinking that glass of orange juice. So if you go to juice, you're increasing the amount of sulfur getting into your body by a significant percentage. So if you've started juicing and you're not feeling well and you notice that you have these CBS SNPs, maybe the juicing isn't as healthy for you as you thought it was. COMT, catechol methyltransferase, breaks down excitatory neurotransmitters, those things like epinephrine, norepinephrine, dopamine, glutamate, so forth and so on. Diamine oxidase is what breaks down extracellular histamine. The folate receptors, DHFR, FOL1, if you see polymorphisms here, this is a warning for you not to take things that are going to block the folate receptors. Because if you block the folate receptors, natural folate can't even start the whole process, that whole folate, methylation, methionine, all those pathways that start with folate. Things that block folate are folic acid, believe it or not, ECGC from green tea, dairy, methotrexate, and grapefruit seed extract, which is used a lot of times in yeast treatment, so be careful. FUT2, fucosyl transferase 2. I can never pronounce it because I always think I'm cursing. This interferes with your ability to hold on to B12. Okay? I've gotten a lot of flack about this one, but that's part of what it is. And there's a lot of research out there that says that FUT2 can also affect the microbiome in the gut. G6PD, glucose 6-phosphate dehydrogenase. If there's a deficiency here, it could cause a lot of havoc in the body. So if you have polymorphisms there, which we don't see here, it would behoove you to get that checked medically. The next set of genes, which we'll go to the next page, I want you to know that a cell wall is a phospholipid bilayer. In all of my lectures, I am forever saying that a cell wall is a phospholipid bilayer. People think I say it because I like the way it sounds. Okay, but one of the base reasons people do not heal is because their cell walls are leaking because there aren't enough phospholipids to repair the phospholipid bilayer. Concerning MTHFR1, this gene has contribution to the methylation pathway and also has indicators in difficulty in storing the phospholipid choline or phosphatidylcholine that's necessary for cell wall repair. Okay, let's get to it. MTHFR, methylene tetrahydrofolate reductase. People, there are 50, count them, 50 MTHFR genes. There's a lot of them here, but remember the medical guys only test MTHFR A1298C and C677T. And God forbid you have one of them, they go absolutely nuts. The reality is that if you have a polymorphism here, you may have a reduced capacity to produce methylfolate. You might have reduced levels of BH4 and SAMe. There's numerous effects from that, including in the biopterin pathway, which is involved in neurotransmitter formation. The variant of the MTHFR doesn't really matter, although there are some variants that have a little bit more powerful reduction in enzyme kinetics, such as the C677T and the A1298C, but combinations of MTHFR SNPs may indicate more restrictions in the pathways. Again, like I told you before, it all has to do with traffic. And this particular pathway, just looking at it this way, I wouldn't be overly concerned. I'd be concerned about other things. If we go to MTHFR, MTHFS, my consistent joke, I know it has meaning, but I'll let you know the takeaway is everybody's got a polymorphism there. And not the entire population is sick, although I know that people would argue with me about that. Okay, but I tend to pay not as much attention to it as I do everything else. MTRR is conversion of methionine, and the MTR is conversion of methionine, and MTRs are conversion to methyl B12. So if there's a lot of polymorphisms there, you might think that this person may need methyl B12. More important, health-wise, are your NOSs, nitrous oxide synthase. Nitrous oxide synthase is what breaks down free radicals. Okay, and again, there's an association with mitochondrial dysfunction. When you see the SODs and the NOSs and there's a lot of polymorphisms, you got to be thinking mitochondrial dysfunction. And here you can see there's more NOSs here. In the Biogerontology 2013, it says the gene variations of NOS1 and NOS2 were associated with longevity. And in addition, the NOS1 was also found to be associated with lower cognitive performance. NOS2 polymorphisms showed to be associated with physical performance. Like I said, what this is really talking about is the fact that you're able to or not to break down your free radicals. And if anybody remembers free radicals, oxidative stress, inflammation, they're all kind of equivalent terms. If anybody's as old as me, you remember the book Life Extension by Dirk Pearson and Sandy Shaw. That's kind of what started the ball rolling when it concerned free radicals. PEMT, phosphatidylethanolamine. This is better evidence that you have difficulty in that phospholipid area because phosphatidylethylamine turns into phosphatidylcholine. And let's face it, this is how you produce your phospholipids for cellular repair. SHMT has to do with, we call it the leaky gut gene. Okay, the SLCs are associated with the folate receptors and basically are carrier ions, carrier enzymes. Okay, and the TCN transcobalamin genes may reduce the transport of B12. The VDRs. Diseases associated with VDR can include osteoporosis, vitamin D-dependent rickets, and there is a whole ton of research out there that we can spend about 3 or 4 hours going through. Any polymorphisms in the celiac area basically just put you on notice that you might be gluten intolerant. Polymorphisms in the thyroid area, possible thyroid problems. See a lot of polymorphisms, you should get the proper thyroid testing done. Eye health. The BCMO is what takes beta carotene and turns it into vitamin A. Somebody who's expressing here will be drinking a lot of carrot juice or eating yams, and they kind of get a tan, okay, because they're not breaking down the beta carotene, okay? That's what that means. Mitochondrial function. Very quickly, our energy is created by glucose. Glucose creates ATP, adenosine triphosphate, which is our energy. 1 mole of glucose, and a mole is not a furry animal, it's a measurement, okay, produces 38 ATPs. But the first thing that has to happen is that glucose has to go through glycolysis, and the product of that is pyruvate or lactate, lactic pyruvic acid, and 2 ATPs. If you have enough oxygen, the next step would go into the Krebs cycle via acetyl coenzyme A. The Krebs cycle is a honking amount of biochemical reactions that will create the electron donors known as NADH and FADH2, which will go into the mitochondrial electron transport chain and produce ATP, the other 36 ATPs. If there's not enough oxygen and you're in an anaerobic state, okay, the products of glycolysis will not go through the Krebs cycle and then just start creating lots of acid, and everything that's bad in the body is acid loving. So that's why we're always talking about alkalinizing, okay, because you want things to go, you want the products to go into the Krebs cycle and experience oxidative phosphorylation. The electron transport chain, this is a cross-section of the mitochondria. This is how we create our energy. For those people who are purists, the mitochondria is not native to our genome. Apparently, way back when, in a galaxy far, far away, there was a bacteria that hung out with our cells, and our waste products was its food, and its waste products was our energy. So a deal was struck, and there was an accord. And now we have the mitochondria, which has its own DNA, okay, and it's got a kind of a funny-looking cell structure because it's got an inner and outer cell wall, and it's got 5 complexes, okay. So in the first complex, NDUFS takes NADH and pulls off the H and puts the proton into the intermembrane space. By the way, these guys are ferried in by coenzyme Q10. And then in complex 2, succinate dehydrogenase pulls the 2 H's off FADH2, holds on to the electrons and throws those protons into the intermembrane space. Then we go through complex 3 and 4, which perform what I like to call magic, because any science sufficiently advanced is indistinguishable from magic. And the end result is they go down to that 5th complex that looks kind of funny on the bottom there, but actually looks like a little factory, and all those pluses, all those hydrogens kind of run that. Okay, and that's how you get your ATPs. And in order to have your energy, not only do you need the results of Krebs, which is NADH and FADH2. You need clear pathways, okay? In other words, 8-lane highways instead of 2-lane highways. You need those cells not to be leaky, otherwise you're going to lose those protons. Okay, and you need all the coenzymes and cofactors to have the reactions work. When they did this, they kind of put it in alphabetical order, not complex order, so I put the complexes next to it. ATP would be complex 5. COX is complex 3. NDUFS is complex 1. And here you can see where the real problem is in complex 1. And UQCRC2 is complex 4. And you know something, I may be mistaken because I recently learned that UQCRC2 may be complex 3 and COX may be complex 4, but frankly it doesn't matter because it's the complex 1 where the issues are. Okay, and in the literature, this is where the problems are. And you notice that instead of having an 8-lane highway, this person has a 2-lane highway. And if there's a lot of oxidative stress, what's going to happen is, and Ben Lynch found this out, okay, what's going to happen is oxidized glutathione, otherwise known as GSSG, it's going to get stuck in that first complex in this area and prevent the entry of the electron donors that will end up sailing through the system to create ATP. So you're not getting your ATP and you cannot heal. Let me digress for just a moment. You know how much I love my digressions. If you remember way back when, coenzyme Q10 was brought here from Japan because the Japanese dentists noticed that they could give their patients coenzyme Q10 and it would cure periodontal disease. Of course, it came to the United States. Multi-level marketing took over and screwed everything up. But the reality was it didn't cure 90% of the people of all their ills. It helped about 30 or 40% of the people, and this is just before we knew that it was part of the mitochondrial pathway. But let's not forget the original reason. Somebody got periodontal disease, and because their diet was bereft of coenzyme Q10 and they weren't producing ATP, they did not heal. That's the takeaway, people. Okay, so you're not producing ATP, you're not going to heal. Your detoxification pathways are all blocked up because of all the traffic, you're not going to heal. If your folate, methylation, methionine, transsulfuration pathway is all blocked up, either from one end or the other or in the middle, you're not going to heal. You need all of this to heal. APOE has to do with Alzheimer's and actually a whole mess of other things. But in this particular case, we're not differentiating the APOE 1, 2, 3, or 4. So that's on the next version, which is presently out. And there's also a very good podcast on my website with Dr. Gondry who explained APOE. The rest of this is not very significant. They talked about familial Mediterranean fever. I don't know if many people that have that. The sulfonyluretransferases are important because it's another indicator of whether you're handling your sulfur activity well. Sulfur is important in the body, but the sulfonyluretransferases are important in the metabolism of certain drugs and endogenous compounds. And actually, if you have CBS and you have other sulfation problems, this will add to it and should be paid attention to. So, now that you've listened to me for the past 30 or 40 minutes, let's tie it together. By now, I hope I've conveyed to you the following. The presence or absence of SNPs in and of themselves do not indicate the presence or absence of disease. SNPs are probabilities and need to be correlated with your entire clinical condition. Treating only the SNPs with various available products designed for the same without correlating your clinical condition is inadvisable at best. And let me just take a moment here that it is February 2015, and I am seeing company after company after company pop up, taking the 23andMe data, running it through something, and handing you a treatment plan that says, hey, I can give you the vitamins that are going to make you healthier. That's not the way these are used, people. Okay, that's not the way these are used. And anybody who does that, you should run far, far away, because by and large, you're going to get hurt, or you're going to spend a lot of money for nothing. This correlation should be done by a trained and experienced healthcare provider. I've always gone by the saying, a doctor who treats himself has got a fool for a patient. This is also, the reverse of this is also true. This is why we're teaching courses now to train the healthcare providers, since the interest is so high, that they should understand how to properly utilize the genetic information for the best benefit of the patient. Okay, a lot of people are waving the methylation flag these days and saying, come on in, I treat MTHFR, I treat methylation. You really have to be careful. You really have to be careful. Discovering what is stressing or crashing the pathways, remember the highway example, is the true way of healing an individual. And often when you do this, the pathways fix themselves. I know that sounds like sacrilege, but believe it or not, if you take away the inflammation, if you take away what's bugging somebody, guess what? They'll start healing. And depending on how long they've been sick and how much damage there is there, okay, they may not need a lot of support in their pathways. May need some support like vitamins and minerals and stuff so that the enzymes will work, but direct intervention into pathways sometimes is unnecessary. So in choosing a healthcare practitioner, it's critically important that you pick someone who thinks like a detective. Very important. A true holistic practitioner will give you the best of traditional and alternative medicine. That's why it's called integrative medicine. How do you know you have a good practitioner? This practitioner will not be beholden to a single protocol or a single way of thinking. The fact is, people, if all you have is a hammer, everything looks like a nail. Your detective will build a treatment plan based around your individual genetics and physiology, and most of all, will listen to you. It's been said by very old doctors, if you listen to your patient, they will tell you what is wrong. Hence, we created bio-individualized medicine. By we, I mean Shawn and myself. Bio-individualized medicine takes genetics and integrative medicine to a new level. By combining the knowledge of neuroendoimmunology, epigenetics, nutrigenomics, acquired or secondary mitochondrial dysfunction, and cell wall integrity, the practitioner trained and experienced in this area has the capability of identifying and treating not only the root causes of dysfunction, but also attending to the downstream effects. That is, the effects that were created by what damaged the body. That is, if we fix the primary causative agent, what the primary causative agent did to the body, as well as fixing the root causes, okay, you're going to create health. But this has to be done on an individual basis, which is why I don't write a book. Each person is different with varied requirements. There are no protocols, people. People call me up and say, do you do a protocol? I say, no. Protocol is a 4-letter word in my office. But practitioners of this paradigm are finding answers that have eluded others and are developing treatments that show promise in eradicating chronic illness and returning patients to normal function. Practitioners trained and experienced in this area include myself, Shawn Bean, and of course Dr. Ben Lynch, and very soon many others. Alyssa's story. Alyssa was 8 years old when her mom contacted me. They were in Texas. Okay, and I do have permission to use her name. And Mom thought that she needed Irlen glasses, which are the glasses that you see her wearing. Irlen syndrome is something called scotopic sensitivity syndrome, where the brain can't handle all the frequencies of light going in, causing visual distortions. But after listening to Mom, it struck me that this was not the only problem. So I had her ask Alyssa a bunch of questions, and the next day Mom called me up very, very upset because Alyssa not only had a bad gut, a bad gastrointestinal system, but also had visual, auditory, and olfactory, meaning smelling, hallucinations. She would smell things that weren't there. Well, the fact is, people, that olfactory hallucinations are a brain tumor unless proven otherwise. So I asked Mom, did she get the workup done? And Mom said, workup? I said, okay, here's what you do. And I sent her to her doctors, and I said, if everything comes back negative, you come back and see me. And she did. Because everything was negative. So here we have a young girl, 8 years old, who on the horizon will be on antipsychotic medication for the rest of her life. That was unacceptable. So we started looking around for the root causes. We started treating her gut by doing leaky gut syndrome, GI protocols. It took us a little while. And I was working with an integrative pediatrician. By and by, we found out that what was bothering Alyssa was 4 different infections, to include Lyme, Anaplasma, yeast, and HHV6. We got her 23andMe, and this was in the early days of it, but even Sterling called me up and said, you got to do something about this girl, because she had so many reds that we knew something was going to be horribly wrong. And therein lies the story. We cleaned up her gut. The integrative pediatrician wanted to then put a PICC line in her and start rotating antibiotics. I disagreed with that vehemently and wanted to go into a different direction, and got with the parents, explained the pros and cons of everything very fully, and the parents chose to go in the direction I wanted to go, with a non-pharmaceutical approach. And we did that for about 6 weeks. Redid the tests, and everything was gone. Plus, Alyssa had no more hallucinations. And interestingly, that's the way it stayed as long as she followed her diet. Even though she had the worst looking 23andMe going, that anybody will look at and say, this person must be sick, because there's no way they can be well. Because of bioindividualized medicine and looking at her individually, she is now almost 12. She's a normal tween, I guess they call it these days. And she does not need to be on antipsychotic medications. So guess what? This stuff does work. All you have to do is listen and think. Consults are available by Skype or phone worldwide. Also, we educate and mentor other healthcare providers in what we teach. I'm the bald guy on the left, and you can email me at bioindividualmed@gmail.com. There's my phone number, 610-449-9716, or you can fill out the contact form at methylationsupport.com. Shawn Bean can be, he's the muscular-looking guy on the right. Guess I should have put names down, huh? His email is info@matrixhealthwell.com. His phone number is 484-868-0916. Or again, you can fill out the contact form at methylationsupport.com. Personally, I offer a complimentary 15-minute get acquainted session to ascertain if I can help your condition. All you have to do is call and schedule that with my staff. I thank you for your time. I hope you learned something tonight, and we look forward to seeing you in the future, listening to your questions, and I bid you good night.